So my name is Jacqueline, and I spent this summer working in the guided therapeutics lab at the Princess Margaret Cancer Research Center with Dr. Michael Daly. And my project is focusing on the impact of multi-balanced light propagation on fluorescence imaging for cancer surgery. So our lab focuses on cancer because nearly 1 in 2 Canadians will actually be diagnosed with cancer in their lifetimes. And specifically, our focus is on head and neck cancers. So head and neck cancer is the sixth most common cancer. It has a 5-year survival rate of only 50%, so relatively poor prognosis. And the majority of patients actually undergo surgery as a treatment option. So what would happen in surgery is that a surgeon would go in and resect a tumor like the one you see on the left, with the aim of actually maintaining as much healthy tissue as possible. So there's a bit of a balance between removing the tumor and keeping healthy tissue. And in surgery, when there are positive margins, meaning that there are tumor cells detected still in the tissue after resection, that can really negatively affect clinical outcomes. About 20% of patients will have positive margins after surgery. So right now, the way that a surgeon is figuring out where the tumor is and how big it is and how to resect it is based off of sort of things which have limited precision. So visual cues, manual palpation, anatomical imaging, and pathology assessment. Our lab is working on new techniques using fluorescence guided surgery to allow for better precision in understanding for the surgeons and improve resections. So the way fluorescence guided surgery works is that you take a contrast agent, which is injected into the body, which will accumulate preferentially at the tumor. And then using a fluorescence imaging system, you can actually image what the tumor looks like to see where the fluorescence is. And that tells you where the tumor is. Now, what my work is focused on is actually improving this imaging system. So the imaging system, as you can see in this image of a rabbit model, sort of tells you where there's fluorescence, where it's bright. But what's often missing is our understanding of how that image is actually affected by lighting variations. So the brightness of the green on this image is not only dependent on the amount of contrast agent, but it can be also affected by the way the light hits the surface. And so in order to gain a more quantitative understanding of what fluorescence means, we have to be able to compensate for those variations. So previously there's this model developed where you can look at how surface distance and angle will affect lighting variations. So it's a bit of a complicated figure, but what I want you to take out of it is that we can use this equation to essentially determine how much light is there at a given point on the surface, and then use that to compensate for the amount of fluorescence that we see. So an example is on the tongue. This is an agar phantom of the tongue. So it's just like an agar model based off of real anatomy. And there's actually consistent fluorescence quantity throughout this whole image. So the raw fluorescence image, you can tell that it's significantly brighter on the anterior side than the posterior side, despite there being the same amount of sort of contrast agent. And so that's actually varying with respect to light. So we can use this model to get that compensated fluorescence image you see on the right, and better understand where there actually is a significant amount of fluorescence. So we also did this in the oral cavity. This is another agar model. So this is a model of the oral cavity, the inside of the mouth. You can see the uvula back there. And we have 3 images here. The first is just the image from an endoscope, so from the camera itself. The second is the direct lighting image. So this is based off of this model, what the image would look like. And then the third is actually a ratio between those 2 images. So it shows where there's discrepancies between the model and the actual image itself. And the thing I want to point out here is one thing that we notice within the oral cavity is there's these large discrepancies in modeling at the back of the mouth. So there's significantly more light here than is expected. And so our hypothesis here is that there's some sort of light funneling effect. So light can come directly from the source and hit the surface, but it can also, once it hits that surface, sort of bounce off and bounce around and actually sort of amplify the intensity of light. So the goal of my project was to look at this multi-bounce lighting effect and understand how does that affect light distribution in the oral cavity. Where is it relevant? Is it something we should be modeling for and considering in our fluorescence imaging systems? So before I move on, I just want to introduce 2 terms that I'm going to be using. The first is direct lighting. So when I'm talking about direct lighting, what I mean is just the light directly from the light source that hits the tissue surface. And then global lighting is the second term. And when I talk about global lighting, what I mean is the light that is actually being reflected between different parts of the surface and the light that's added from that effect. So in order to look at this effect, we had to model it. So I developed a computational model in MATLAB that allowed you to essentially simulate a series of successive bounces of light. So you can imagine one bounce hits, and then you consider how much light is being transferred on the second bounce, etc. And what we had to do for this is have an understanding of how much light is actually transmitted between two faces on the surface. So there's this equation that we can use. It's very similar actually to the direct lighting model equation I showed earlier, except it's also accounting for the fact that some light will be absorbed when it hits the surface. The other thing I had to do in order to develop this model is determine which faces on the surface we're actually considering for these computations. So for the direct lighting, you're just looking at faces which are lit up by the light source, as you can see here. But then the global lighting, we're actually considering a wider range of faces because there's a chance that sort of the light bounces off the top and hits the bottom where it wouldn't otherwise be illuminated, bounces back, etc. So you have to consider a little bit more of a broader range. So using this model, we got a series of images, so simulated images. What I'm showing here on the left and the right, the left is just sort of the simulation of what the direct lighting should look like. And then the right is how much light would be added by global lighting. So they're on the same scale. So you can see sort of how much quantity of light is added by global lighting. And the thing I want to point out is that we see, again, sort of how I showed way earlier, that you have that discrepancy at the back of the mouth. It's indicated here based off this modeling that it's actually the areas near the back of the mouth that are showing that larger increase in light intensity. And in this case, for instance, it's up to sort of like an 80% increase in light intensity, which is pretty significant. So if we look across all 30 images, this is information about the global lighting images. What you see is that on average across the image, the light intensity is increasing about 15%. But if you're looking at specific regions, it's much more significant. So the key takeaway here is that global lighting actually increases maximum lighting by an average of 50%. So there's specific areas in which this has a much greater effect, like we can see on that image. And that's something that's really important for understanding in what context this would actually be important to model and important to understand. So the next thing we did is took these computational models and these simulated images and compared them back to those endoscopic images, the series that we had before. And you can see here, the top row is the direct lighting model, and then the bottom row is the global lighting model. And there's greater agreement in the ratio images with the global lighting model. You no longer see that sort of effect of light funneling at the back of the mouth. It's actually a lot closer to 1, which is what's desired. Then we took a look at all 30 images. So on the left side, I have the mean of the pixel by pixel ratios. So basically the mean of all those ratio images. And what you can see on this graph is that with the direct lighting model, the mean of those ratios is around 1.5, 1.6. But when you look at the global lighting model, it's a lot closer to 1. So there's much more significant agreement between the simulation and the actual image. And then if you're looking at the standard deviation, the thing I want to point out here is just that the global model has less deviation, which is suggesting that there isn't some of those areas where there's huge discrepancies. Those might be accounted for by this global lighting model. So in terms of next steps, the 3 I have down here are first really understanding what impact this has on sort of tumor background ratio and on actual clinical scenarios. Maybe in some cases, if you're looking at a tumor at the front of the mouth, it's not actually relevant, but it's important to understand what its application is specifically in clinical scenarios. The second is that this can apply to other types of cancer surgery, maybe in the brain, lung, abdominal tissue, anywhere where you have high curvature surface that's relatively reflective. And then third is clinical translation to ongoing patient studies, because we want to be able to understand not only what's the theory of this, but how does it actually affect application and can it improve clinical outcomes? So in summary, global lighting plays a significant role in light propagation in the oral cavity, and this data might be relevant to other structures and imaging applications. just want to thank my supervisor, Dr. Michael Daly, as well as the funding support that we got for this project. And that's it for me. Thank you.
Thank you, Jacqueline. So, Simon, if you would like to unmute yourself.
Yeah, like really great presentation. It's interesting this kind of like analysis of the global lighting. Do you think that, because you said that it's transferable to other like model of cancer, do you think that the global equation will stay the same, or is it going to be like completely different? Because you can assume that the surface, like if you look on the lung, there will be some like mucus, some different cell types, so maybe like the absorption, the reflection will not be the same. So do you think how different it's going to be like in your mind?
Yeah, so the equation right now, essentially the one term that could be changed by like a different tissue is it's using like the diffuse reflectance, which is basically telling you how much light is being reflected off the surface. So if you're in a tissue that has a lot higher absorption, there would be it would be a lower diffuse reflectance. And so like you'd probably observe less of these effects. Whereas if you're in something with like maybe like higher scattering properties and like basically more reflectance, then you probably observe these effects more. But the equation itself should remain the same because it's based off of like Lambertian-like dispersion, which should remain the same in different tissues.
Okay, yeah. Thank you.
Hello, Jacqueline. This is Sharianne here. Thank you for a very enlightening presentation. My question is with respect to the direct modeling and the global modeling. Given the discrepancies that we saw in your presentation, do you think there's an application for previous diagnoses to be changed and treatment as such to be changed because of some of the light, because of the global modeling, some of the light being reflected is duplicated? Do you think that application in terms of going back to previous patients, do you think that's something that's going to be looked at?
I think in this case, so like in the case of head and neck cancer, fluorescence imaging is something where right now they're sort of in clinical trials for the fluorescent contrast agent. So it hasn't necessarily been implemented and those decisions haven't been made. I think the thing about this modeling is that it really can be like pretty structure specific. So if you're looking at, obviously we saw the example of the oral cavity, it might be like a significant impact here. But if something's sort of like a flat surface, it wouldn't necessarily affect it. So I really don't necessarily know enough about where like lighting models like this are being applied to say whether that would change things in that respect. I don't know if that sort of answers your question.
Sure, yes. I was just wondering if it's something you had considered, given the yes, I totally understand based on where the light is shone that reflections can be different. I just thought that it could help with previously misdiagnosed patients in terms of possibly reducing treatment. So thank you.
Yeah, I think it would help. Like the idea is it would just like increase precision going forward.
Right.
For like some of these resections. Oh, great question. Thank you.
Thank you, Jacqueline. Are there any more questions?